Perfect. All right. Thanks, Dr. Matt. Well, good afternoon, everyone. My name is John Bodie. I am the Vice President of Operations for the Mold Pros. And after that great presentation from Dr. Lauren, we're going to talk about the environmental side of environmental illness. Um, I am a certified indoor environmental professional licensed by the state of Florida, Louisiana, and Texas. Work for the Mold Pros, which is a national environmental services firm with locations throughout the U.S. We specialize in assessments and remediation for properties for those that have environmental illness. Our agenda today, we're going to talk about property inspection for those that have environmental illness. I understand we have a fair amount of IEPs on the call or webinar today, which is exciting. Um, the, the road to wellness for a lot of our clients and a lot of our doctors' patients begins with a proper inspection. So happy to share about that. Then we'll talk about some different types of sampling. Oftentimes we get questioned on what's the best type of sampling. Should we use this one versus that one and that sort of thing. So we'll try to unpack a lot of that today. We'll talk primarily about the two main types of sampling or laboratory analysis, which is direct microscopic exam. And we'll talk about some PCR sampling, including the Hertz-Me2. We'll talk about the ERMI test and the EMA test. And then we'll talk about the happy path when everything kind of lines up and we get the results that we hope to see from the clinical and environmental side. And then we'll talk also about sampling protocols on how to offer pre and post treatment. So oftentimes I get asked, hey, John, is a professional assessment really necessary? And not every time do we say an assessment's required. However, there are times when we feel that a professional assessment is required, and that's really when it boils down to a health concern for the occupants. The other time is when we have that clinical testing, which indicates there's been exposures to fungal contaminations or other toxic contaminations. So I wanted to talk to our IEP friends that are on today's webinar, as well as maybe some homeowners and some of our healthcare practitioners that are still on. The main job of a mold inspector is to determine the fungal ecology of a structure. And let's talk a little bit about what's the fungal ecology of a structure. It's there's always going to be mold inside of a home. Every time we open up a door, a window, we have air exchange, etc. It's always going to be there. So what we want to understand is what is normally inside that structure when compared to the natural environment outside of the structure and determine if there's any elevations. So an inspector's main job, a professional inspection should identify several things: the what, the where, the why, and lastly, the how. And we'll talk about that. So the what, we should really understand, and this is where our sampling comes in. A professional assessment should indicate the what type and what amount of contamination is inside of the structure. It should also identify the where. Is it contained to a certain area of the house? Is it just in the basement, the crawl space, the living space, attic, etc.? And that's really where that visual assessment comes in. And again, the sampling, breaking the house down into zones and taking proper samples. That's critical to determine the where. And then the why. And this is really where a lot of your assessments should begin, is that's the moisture. We'll talk more about that on the next slide, is why is it there? Worst case scenario for a homeowner is to have someone come out, assess, understand that there is an issue, but not know why it's there, and wind up having to deal with the same thing all over again. And then the last thing is the how. So after we get all the data back from the laboratory, the assessment's been done, the visual side of things, all the fungal, any other testing that we've done, all that comes back. It is the job of a professional IEP to write a protocol to return that property back to a normal fungal ecology, and that's the how. So after doing this for about 10 years plus, all of our assessments begin, we call it the 3 M's: moisture, mold, and then mycotoxins. And as I alluded to on the last slide, A critical thing is taking the time to visually assess the property. An assessment should not be a 30 or 45-minute air sample and then done. The assessment should begin from the exterior of the home, looking for grading issues, flashing issues around chimneys, that sort of thing, open penetrations into the home, etc., and work its way from the exterior into the interior. And then on the interior, we should work ourselves from foundation to rafters, inspecting the entire property. One of the things as an IEP, we all get talked about or trained on water damaged buildings, and we're keen to look in wet rooms, kitchens, and bathrooms, and laundry rooms, and those sorts of things. But I would encourage you to also spend the time to look at personal contents. There's a lot more surface space that's there, and I think that can be a huge differentiating factor, especially when we have xerophilic molds, is by spending the time to look at personal contents. And by that, I mean looking with a high lumen flashlight at nightstands or the back of headboards or underneath dining tables, pool tables, pianos. Those are oftentimes places where xerophilic molds such as penicillium or aspergillus can attach themselves and start growing due to the restricted airflow that's there. We're trained, of course, to look for discolorations in ceilings. Also pay attention to heavy dust levels. Of course, water stains and HVAC systems, which we'll talk about. A good inspection should include all of these factors. And the best thing to do, oftentimes we have some pretty nice tools in our tool chest, but it's the eyes and the nose, which are some of the best tools that you have. Smelling for VOCs as soon as you walk in, and taking the time using the high lumen flashlight. Actually, we use black lights as well whenever we have interior surfaces that are lighter in color. For example, walls that it's really difficult to see aspergillus on those light color walls. High lumen flashlight or black light can help get that vegetative growth to pop. Other tools that are in the quiver of an IEP, infrared thermography to help to find moisture issues or thermal anomalies inside of a structure. As a homeowner, you should look for an IEP that uses these tools in addition to moisture meters, borescopes, et cetera, to find hidden issues which aren't readily apparent to the eye. HVAC systems. An inspection is not a complete inspection without a thorough inspection of the HVAC system. The HVAC, which stands for heating, ventilation, and air conditioning, is essentially the lungs of the structure. All the air goes through it. It can either be our best friend if it's clean and we have great filtration and circulating air. It can help us out in maintaining a normal fungal ecology. And of course, if it's not, and it's not pulling out the moisture in the air as it should, or if it's contaminated, then we're going to wind up having issues. And it can actually be our worst enemy. So whenever we're doing an inspection, we talked a little bit about the humidity levels. You should take humidity levels or readings all throughout the structure, and especially paying attention to the HVAC vents. I live in South Florida. We have a lot of issues, of course, with flex duct and all the new construction. We'll wind up getting the heat. We'll have the adhesive on the flex duct will separate, or we'll get palmetto rats or something along those lines that will may chew holes, or there's tears or penetrations in that flex duct. So that can actually introduce contaminated and certainly elevated moisture air from the attic into the structure. If you're not testing the air coming out of your HVAC vents, it's really difficult to understand where elevated humidity is coming from. So we encourage you to certainly take a look at that. Then once you've done your inspection of a home, then at that point, it's a matter of setting back and saying, okay, what type of samples are needed? Is it an air sample? Is it a surface sample? Is it a PCR sample? At that point, get your clients buy-in, understanding, and approval for all the samples. Take the samples needed, send them off to the lab. Once you get the results back, then turn around and build your protocol based upon the laboratory and the visual findings. One of the things that we always encourage folks to do, and it seems like a simple thing, but let's rely on the science and make really good data-driven decisions. So oftentimes, a popular question I know we all get is, "Hey, what's an acceptable level of mold inside of a structure?" And the EPA states that there are no threshold limits set for airborne contaminations. So it really is reliant upon if you're trying to understand what's acceptable and what's not, or if there's elevations or if there are no elevations. If you're doing an air sample, that's comparing it to the exterior baseline and doing a compare and contrast. And I thought this was interesting for the World Health Organization. It states that microbial growth may result in greater number of spores, cell fragments, allergens, mycotoxins, endotoxins, which of course are bacterial toxins, mycotoxins, as we know, are mold toxins, beta glucans, and volatile organic compounds in indoor air. The causative agents of adverse health effects have not been identified conclusively, but an excess level of any of these agents in an indoor environment is a potential health hazard. So as an IEP, this is really telling us what things we should be really looking for from a mycotoxin, endotoxin, fungal standpoint, bacterial standpoint, and of course VOCs. So environmental sampling, it really breaks down to, as we mentioned, two types. We have direct microscopic exam, which is going to be at the genus level, which is, again, live in South Florida, so I use a fishing analogy. I would say that a direct microscopic exam is like casting a broad net. You throw it out there, whatever comes back is what comes back is what you get. And we'll talk a little bit about spore traps. We do a lot of intracavity samples. If there's water damage on building materials and drywall, we certainly want to understand what's going on inside that drywall without being destructive. So if you're not doing intracavity samples, that's a great tool. It's easy to do. It's not that invasive for a homeowner and can really provide some insights. We do a lot of tape samples. When there's visible fungal growth or suspected microbial growth, you should be doing a tape sample where that SMG is. And of course, a swab sample is to be used if you cannot get adhesion on a tape and it's wet or it's in a hard to reach place, you can use a swab. In our practice, what we see a lot of these days are auger plates. I'll touch briefly on auger plates. Don't use them too much in the IEP world. They're gaining in popularity. You can find them at all your lumber stores or home improvement stores. And so we'll talk about the pros and cons of some auger plates. And if you're not doing VOC sampling and you want to serve those that have environmental illness, I would strongly suggest you get trained and take a look at that. We do work for a lot of patients that have Lyme disease, and a lot of those individuals are chemically sensitive, and it's helpful to have that data point. The top line here, I think, is really important. It's proper sampling is key to understanding the fungal ecology of a structure. Multiple sampling methodologies should be used. There isn't one panacea or one magic bullet. It's going to be a combination of all the tools that we have in our quiver, which really help us understand what's going on inside that home. And then in conjunction with direct microscopic exam, we have PCR or mold-specific quantitative polymerase chain reaction. Yeah, it's a mouthful, but that's exactly what it is when it comes to doing DNA sampling. A couple of tests that we'll talk about today. We'll talk about the ERMI test, the HERTS-Me2 for healthcare providers that follow a shoemaker protocol. You'll see a lot of the HERTS-Me2 test out there. And of course, the EMA, a lot of our functional medicine physicians will use the EMA test, which actually looks for molds and mycotoxins. And all of these can be collected with a dust sample, whether it's vacuum or bulk or swab. Or my favorite is using an electrostatic Swiffer. So we'll talk more about all of those. But first, let's talk about spore trap. Probably the most common sample that gets used in our industry today. Been around forever. It has some strengths and weaknesses, and we'll speak to that in a little bit. but it's used really in the inhalation zone. So you come in, you take an exterior baseline sample to understand what the fungal ecology is on the outside, and then you're going to take a sample on the inside. Each sample takes in about 1,000 to 1,250 square feet, depending upon the layout and the airflow of the home. So it does a pretty good job of capturing the majority of the spores or particulate matter in the air. It can be used not just for mold testing, but when we have clients that have allergen issues, you can run pollen panels for elm or oak or grasses or those sorts of things. It'll pick up other items such as skin cells, insect fragments, fiberglass, and larger high full fragments as well. Um, so it gives us, if you're taking your sample 3 to 6 feet off of the ground, which is where you should be taking it, so it helps you understand what that inhalation risk of the occupants is. Um, and again, it's always a compare and contrast. Sometimes we'll see indoor air samples with no exterior baseline, and so it doesn't really tell you if that structure or that property is elevated or not. You have to compare it to an exterior baseline. It is subject to exterior weather outside, and that's one of the downsides, and we'll talk a little bit more about that. So some of the disadvantages of spore trap testing. Certain molds underneath a microscope, they look the same. An example of that would be Penicillium and Aspergillus. And so these molds will get lumped together. They're not broken out, and it's always at the genus level. So the other thing is that molds have sporulation cycles, just like flowers have blooming cycles. And so if you're doing your sampling that day, your sampling, of course, is a snapshot in time. You could be off cycle on a sporulation, and therefore it's underrepresented on what you have. And so that's something you have to consider. You also have occlusion on the lab slide. And essentially what occlusion on the lab slide is when there's heavy particulate matter, whether that's other spores or maybe fiberglass or insulation or dust, covers up that lab slide. When the technician looks at it underneath the microscope, you can't make out and count all the mold spores that are there or differentiate, and therefore you're going to get a negative bias on your results. And the exterior baseline can be influenced by outside environmental factors. Snowpack, for those that are in northern climates that get snow, it'll encapsulate the spores, and it'll wind up looking lower on the outside than what it typically is, and therefore make your interior counts look higher than what they probably are relative to the exterior baseline. Also, rain. Rain will, when you have lighter spores, xerophilic molds like Aspergillus and Penicillium, the rain will wash them out. And so therefore your exterior baseline will have less pinasp than what it would otherwise. Conversely, when you have molds that actually sporulate during rain cycles like basidiospores, you're going to wind up having an elevation of basidiospores on your sample. So just be aware that your exterior baseline can be influenced by the environment. And then the other thing that I see quite commonly is that most IEPs don't take the time or haven't been trained to actually understand that spore trap result. One of the great things to look at is the relative distribution of bioaerosols, in other words, the percentages. So we should have, in a normal fungal ecology, the same molds on the outside and the same percentages as what we have on the inside. And the counts, and certainly there should be less on the inside than the exterior, of course. But look at those percentages. And also factor in the layout of the structure. If you have where your egress, if it's at the front of the house and we have an irregular distribution of bioaerosols in, say, the master bedroom where there's no egress, then you have to look at that and make that interpretation of the data. And then, as we mentioned, surface samples. And in my opinion, a surface sample always trumps an air sample because of those sporulation cycles, et cetera. So whenever we see vegetative growth, we should recommend a surface sample. We do a lot of tape lifts, and as I mentioned, swabs can be effective when there's hard to reach places or on wet surfaces. If you're sending in your swabs, you should be, and they're damp, you should be cold packing those. That way you're not creating a cultured sample or growing mold while you're sending it off to the lab. You could also use a bulk sample. A 2x2 or 3x3 section of wood or drywall is helpful. And again, this analysis is going to be at the genus level again, as with other microscopic exams. It doesn't identify the species. But it does a good job when we have species or types of molds like Stachybotrys, which don't aerosolize, that you would not miss, that you would miss and not capture during a typical spore trap. You will capture it with your tape samples. So that's a huge advantage. And again, the same disadvantages. It's not going to tell you is it Aspergillus or Penicillium, or it's certainly not going to tell you if it's a toxic or pathogenic fungi at all. Auger plates. We're seeing more and more auger plates being used, and typically these auger plates, they're going to be filled with like a potato dextrose or sugar of some sort, which will allow for rapid growth of molds. Their advantages are they're extremely inexpensive. You can pick them up at any of your big box home improvement stores, and they're pretty easy for a consumer to use. It doesn't require any expensive equipment. And auger plates are good, or they should be used if you're trying to determine if we have viable molds or living spores that can grow. That's really the strong suit. The disadvantages of using auger plates, and we have to be prepared to have this talk track with a lot of our homeowners that do these DIY kits, is that there's a lot of variability. So, how much airflow crossed over that auger plate. You have no idea what volume is. Oftentimes, you don't capture the exact amount of time it was out. So we have X amount of fungal growth based on what. So it only tells really a small part of the story. We typically don't use them, but it's a talk track that you need to be able to have and understand so you can help your homeowners out with them. So I think this is an interesting comment. It talks about identification of mold species is vital for studies on mycotoxin production for indoor molds. Chemotaxonomy based on the species-specific production of metabolites is an important tool that can help exclude problems caused by incorrect identification based upon microscopy, contamination of cultures, or erroneous claims of mycotoxin production. So a couple things just to remember. We have sometimes clients that'll say, hey, listen, I have Aspergillus, or I have Penicillium, or I have maybe even Cotomium or Stachybotrys. If you're doing a direct microscopic exam, there's a lot of different types of Aspergillus. They may not be producing or may not be capable of producing toxins, much less producing them. And if you want to understand if we have those toxigenic species, you have to go to PCR DNA. And I think that's critical when we're dealing with folks that have environmental illness to get down to that level. So PCR testing, some of the strengths and advantages, extremely sensitive. I'm a big fan of PCR testing for sure. It really gets down to the level that we need to understand. It doesn't distinguish between spores or hyphal fragments or any other biomass. It just knows how much DNA of that particular species is there. So you're going to see your data come out in spore equivalency, not actual spores. This also makes, if you're doing post testing on a remediation on PCR, you get rid of all the spores or the fungal growth perhaps, but if you don't do a really good job in treating those hyphal fragments or the biomass, you're going to wind up being elevated on your post test, and it's going to create some challenges for you. So again, it identifies the species level, which is better attribution to the mycotoxin production or possible mycotoxin production. And it doesn't require any special equipment. And so it's, with the exception of the molecular entrapment, which we can talk about offline, really easy for a consumer to sample on their own. The disadvantages is it does not distinguish between living or dead fungi. DNA is DNA, and so it'll pick that up. And it's very narrow. It's a deep test, but it's a very narrow test. So if you don't put in the DNA probe for the exact species that you're looking for, it's not going to capture it. And of course, the lab has to pay for each of those DNA probes. So it is not an inexpensive test to run, much more expensive than direct microscopic exams. So some of the most common PCR tests, again, the ERMI, which stands for the Environmental Relative Moldiness Index. A lot of DIY clients will send these ERMI tests in. And so if you're not familiar with how to read the ERMI test or the background on the ERMI test, reach out to me and I can certainly walk you through it. There's some great white papers on it. We can get to you as well. And then there's the Hurts Me Too, which stands for the health effects roster of type specific formers of mycotoxins and inflammagen. And the 2 stands for the second version. That is a mouthful. So anyway, the Hurts Me Too by Dr. Shoemaker. We'll talk more about that. Then the EMA, the Environmental Mold and Mycotoxin Assay, we'll talk more about that. And molecular entrapment. For those of you familiar with the ERMI, think of a airborne PCR test with an exterior baseline. So it's a hybrid test, bleeding edge when it comes to an ERMI type of test. And I can get more information on that if you have questions afterwards. The ERMI. So I'm sure we're all well familiar with it. It was a study, healthy home study that was done, an average really of 1,096 homes. It contains 36 mold species divided in 2 groups. Group 1 was a history of homes that has a history of water damage or visible mold. That's 26 species in there. And then group 2 was a group of homes with no history of water damage or no visible mold. There's 10 species there. And the ERMI comes out with a score, and we don't pay a whole lot of attention to the score. This is nonsensical, in my opinion, in a lot of ways. You can come down with a 0 score, which is great, is where you want to be, by either being really clean, hospital room clean, or you could be extremely moldy in group 2. And since you're subtracting out your totals from group 2, you'll wind up with a 0 negative score by being super moldy. And so we don't pay attention to the score, but the data is really good data. And while this group of molds, 36 species, there's 10 species of Aspergillus there. So you have about 5% of all Aspergillus species represented on the ERMI. It does provide some good insights there. But also, those DNA probes are expensive. So as a homeowner, you're paying for species level data that you may not need. And I would offer up Cladosporium species. I don't know of any pathogenic or toxigenic problems with Cladosporium for humans, but you're paying for that species level analysis, which you may not need. On the right-hand side, this is a paper that was issued by the EPA. It still stands today about the public making indoor mold cleanup decisions based upon this EPA research tool. So it really wasn't designed for making remediation decisions, but it can be a good starting point for some folks. The Hurts Me Too is, as I mentioned, the health effects roster of type-specific formers of mycotoxins and inflammagen. Dr. Shoemaker developed this really a scoring algorithm for individuals with SIRS. It's a very narrow test. It only identifies 5 fungal species, and those 5 are listed there. On this test, of course, 10 or below is acceptable. 11 to 15 is kind of your gray area, which you need a further investigation. And above 15, he recommends that the occupants no longer occupy the structure. So it does really, again, zone in or rifle in on these 5 species. There's only 5 DNA probes. So it's fairly inexpensive from a PCR standpoint to run. So the cost can be much less than a 36 panel ERMI test. But the disadvantages are, again, it's even more narrow. And even though it lists mycotoxins in the name, it doesn't tell you there's mycotoxin production present at all. And I thought this was interesting. This is something from the CDC you can take a look at. It says there's about 180 species of penicillium, or excuse me, of Aspergillus, and fewer than 40 of them are known to cause infections in humans. Aspergillus fumigatus is the most common cause of human Aspergillus infections. Others include, Flavus, Tereus, and Niger. But you'll notice there isn't, this particular test doesn't test for any of those Aspergillus species. Here's another comment from the experts. This is the Institute of Medicine of the National Academy of Sciences, and it talks about in vitro and vivo studies have demonstrated adverse effects, including immunotoxic, neurologic, respiratory, and dermal responses after exposures to specific toxins, bacteria, molds, and their products. Such studies have established that exposure to microbial toxins can occur via inhalation, dermal exposure, and through ingestion of contaminated food. Dr. Lauren touched on this in her presentation as well. So let's remember that. It's through inhalation, dermal exposure, and through ingestion. Those are our three pathways. So as we're thinking about testing and what we should test and how we should test, we'll keep those three things in mind. When we're dealing with folks that have environmental illness, there are some questions that I think are critical for us to answer. So are the organisms found inside the home pathogenic, which is disease causing, or toxinogenic? And what, if any, mycotoxins are being produced? And then, of course, what's the source of the patient's exposure? Does it come from diet? Is it environmental? Those are important questions that need to get answered. There's a study, which, by the way, I can get these links to you afterwards. Just send me an email and I can get them over to you. It talks about metabolite production is influenced by medium composition, temperature, and water activity, indicating that molds are likely to generate different metabolites when they grow on building materials. So just because a species, say Fumigatus or Stachybotrys chartrum, is capable of producing mycotoxins, doesn't mean it's actually produced in them. It may not. It's the environmental conditions which are a factor in that metabolite production. So that leads us to an EMA test. And this is a test that at the Mold Pros we use for all of our clients when we're doing a test for someone that has environmental illness. So it's comprised of two parts. It has a PCR testing for the 10 most pathogenic fungal species. And then it tests for 16 mycotoxins. So actually test for the mycotoxins present inside the structure. And that happy path, a lot of our clients will send to us, or we do work for 60 or 75 different healthcare providers nationwide. We'll get a copy of the clinical. And happy path is, hey, in the urine, we're seeing OTAs or aflatoxin. And in the home, we're seeing aflatoxin or ochratoxin A. And so therefore, we have a good understanding of what that exposure pathway is. And I don't know of any other way of getting to it besides doing an EMA test. So unlike the ERMI or the HERTS-Me 2, the EMA actually detects the presence of mycotoxins, quantifies it by parts per billion. And it breaks down, and we'll show you on the next slide, the actual Aspergillus species, which toxins are associated with it. Very, very easy for a homeowner to do. In fact, I know that the lab actually sends a lot of tests out directly. A client can actually order this test, do it themselves, and understand what their toxic load is inside of a structure, and then call in an IEP to help find out the why. That happens quite a bit. Here's the species which are tested and the associated mycotoxins produced. This comes from the lab itself. Great information there. And then testing. I get asked all the time, where should we test? A lot of clients who will test underneath the refrigerator, which is really not a great way of testing. We want to test, we understand that the three ways of getting that exposure is going to be through dermal contact, inhalation, or ingestion. So if we want to understand inhalation risk, one of the best ways is to sample from the HVAC filter. And this is good for a couple of different ways. If we go through and we sample, or homeowner samples multiple locations, we could be introducing sampling bias. Either we hit a spot that's just really super dusty, like underneath the refrigerator, which hasn't been cleaned out for 3 or 4 years, or dusted from underneath 3 or 4 years, or you hit a spot on a shelf that may have months and months and months and months and months of dust buildup, and it may not really give us an indication of what the inhalation risk is. By sampling from the HVAC filter, that does that for us. And typically, if the homeowner is staying on top of or swapping out their filters, that 1 inch filter is a 90-day filter. It gives you line of sight to what's happened in the last 90 days, so a little more relevant. And we think it's a great data point. There are homes. We have markets in Denver and of course Portland and Seattle. They don't have central heat and air, so at that point, then we turn around and recommend sampling from, say, maybe on top of a refrigerator or ceiling fans or bookshelves, on top of TVs, things that get dusted, relatively frequently. Definitely don't want to sample below the knees. You don't want to sample stuff that gets tracked in. Um, the kit, if you're using a real-time labs EMA kit, comes with sterile medical cotton swabs. We may supplement that with electrostatic Swiffer that's unscented to make sure that we get good dust collection. Then you complete the chain of custody, you send it in, and you get your results back in about 2 weeks or less. So kind of a summary for sampling, and then we'll open it up to some questions. Um, it's a combination of sampling. We understand when we have clients that are down this journey for environmental illness, it's a long journey. It's not an inexpensive journey. And so really making sure we're being good stewards with our clients' money, I think, is important. So we use a combination of things: direct microscopic exams to understand what the fungal ecology is, and it's going to be a combination of what we've talked about. And then we'll supplement that with PCR testing. And of all the tests that are available, the one that we use most common is going to be the EMA, because it tells us toxigenic, pathogenic species, as well as what's going on from a mycotoxin standpoint. So that gives us a really good overview. If you have additional questions that we can't get to today, my contact information is down there below. And then Paul Limmel. Paul's a great resource at Real Time Labs from a knowledge standpoint, and also getting kits out and that sort of thing. So happy to help out in any way that we can. We spend a lot of time talking to our peers on how to best serve this community, which is really underserved, in my opinion, by the restoration and the IEP community as a whole. So very good. Well, it's Matt, if we can open it up for questions.